Hello friends, today we are going to start our new topic in biochemistry, that is fatty acid, cholesterol and bile acid synthesis. We will understand it thoroughly. First of all, overview. Fed state. In fed state, you know that our glucose level is high, so insulin level increases to reduce the glucose level. So insulin by glucagon ratio is more as in fed state. Now we will talk about what actually happens here. Actually, excess carbohydrate is intake during fed state. So during glycolysis, excess carbohydrate is converted to pyruvate, and pyruvate in presence of pyruvate dehydrogenase converted to acetyl coenzyme A. Acetyl coenzyme A help in formation of fatty acid synthesis and the cholesterol. Cholesterol is help in formation of steroid hormone, vitamin D, and bile acid. And now we'll talk about fatty acid synthesis. Fatty acid synthesis is actually de novo synthesis. In this, we'll talk about de novo synthesis, that is lipogenesis. Lipogenesis, de novo means formation from new. Formation from new, that is de novo synthesis. New, we are forming new. Another is discovery. Discoverer is Fyodor Linin, also known as Linin spiral. Fatty acid synthesis also known as Linin spiral. Now we'll talk about fatty acid synthesis. For fatty acid synthesis, the site is organs, liver, adipose tissue, kidney, brain, lactating mammary gland. Our site is liver. For synthesis of fatty acid, site is liver, kidney, adipose tissue, brain and lactating mammary gland. Now we'll talk about organelle, that is cytoplasm. Organelle, where it happens in cytoplasm. Substrate is acetyl coenzyme A. Substrate is acetyl coenzyme A. Source of acetyl coenzyme A is pyruvate. In presence of pyruvate dehydrogenase, it is converted to acetyl coenzyme A. Now we'll talk about the steps. First of all, transportation of acetyl coenzyme A via citrate. So acetyl coenzyme A is formed via citrate in the presence of citrate lyase. Actually, what happened in mitochondria, pyruvate is converted to acetyl coenzyme A in presence of pyruvate dehydrogenase, and acetyl coenzyme A is converted to citrate in presence of citrate synthase and oxaloacetate. Now the acetyl coenzyme A is 2 carbon, oxaloacetate is 4 carbon, it is converted to citrate, that is 6 carbon. And citrate moves out from the mitochondria by the enzyme tricarboxylic acid transporter, TCA transport, tricarboxylic acid transporter. Example citrate को ये help करते, this helps. So citrate is 6 carbon compound in presence of citrate lyase converted to, and acetyl oxaloacetate is being removed, it is converted to acetyl coenzyme A, that is 2 carbon compound. Now acetyl coenzyme A carboxylase, second step is acetyl coenzyme A carboxylase. Acetyl coenzyme, that is 2 carbon, is converted to manual coenzyme A, that is 3 carbon, in presence of A ATP and B biotin and CO2. It is converted to malonyl coenzyme A. This is the rate limiting step in. So acetyl coenzyme carboxylase is the rate limiting enzyme. So we will again see second step, that is acetyl coenzyme carboxylase. Acetyl coenzyme A, that is 2 carbon compound, is converted to malonyl coenzyme A in presence of ATP, biotin and CO2. Now third we will talk about the fatty acid synthesis, that is FAS complex, synthase complex. It is multifunctional enzyme complex. Same polypeptide has a multiple enzyme activity. It has multiple enzyme activity. This is except like a structure. It is homodimer from mat cas DH KR. Yeah, it is homodimer. Each monomer contains 6 enzyme activity. It has 6 enzyme activity. Now we'll see the condensing unit. This is the below, this is 4 is condensing unit, that is MAT and KS, malonyl acetyl transcyclase and ketoacyl synthase. Another is TE is releasing unit, that is thioesterase. Now we'll see about reduction unit. Ketoacyl reductase, dehydrogenase, enol reductase and acyl carrier protein, that is ACP. ACP for human, not it is ACP. So now this is fatty acid synthase complex. Now we'll see about reaction by fatty acid synthase complex. First step is condensation, second is reduction, third is regeneration. We have to regenerate to form a 16 carbon compound. Another is releasing. So first of all, we will see the condensation step. As we know that this is the this has symmetry about this side, that is why it is drawn by green, and this is yellow. I have drawn this. So fatty acid synthase complex in presence of acetyl and manolyl. Acetyl is 2 carbon, manolyl is 3, and in presence of MAT, that is manolyl acetyl transcyclase, it is converted to acetyl. This compound is there, that is fatty acid synthase complex, acetyl 2 carbon, manolyl 3 carbon. This goes here in presence of K, is that is ketoacyl synthase. The next product is formed is fatty acid synthase complex along with the acyl. The product form is ketoacyl, that is 4 carbon here attached. Second step is reduction. In reduction, ketoacyl 4 carbon compound is converted to hydroxyacyl in presence of ketoacyl reductase in NADPH. Where there is reductase, we use there is NADPH in converted to NADP plus. NADPH is converted to NAD plus. Where there is reductase, NADPH converted to NAD plus. Now the hydroxyl acyl is formed. So now in presence of dehydratase, H2O is removed. Now the compound is formed. Enol reduced unsaturated acyl, enol residue is formed. And now what is formed? Enol residue is formed, that is unsaturated acyl residue in presence of ER. In presence of ER, it is converted to acyl, that is 4 carbon compound. Now our third step is repeat condensation and reduction step. That is, as actually what we are now repeating these steps. That is again, that is in presence of acyl, manolyl will form in ketoacyl synthase. This is we saw this is acyl is formed. Then ketoacyl is formed in presence of ketoacyl reductase, dehydrogenase and enol reductase converted to the 6 carbon compound. This again after by multiple repeating and condensation unit of 16 6 to 8 is formed, 8 to 10, 10 to 12, palmitoyl 16 carbon compound is formed. Now the time has come for releasing coenzyme A synthase in presence of H2O is converted to this is gone, and now palmitoyl coenzyme is formed. We have to form a 16 carbon compound, so this is formed. Now we will discuss this all, but first of all, if in exam this question come about fatty acid synthase, how we will write this in so we will write fatty acid synthase. 3 stages are there. For what are the stages? Transport of acetyl coenzyme A to acetyl coenzyme A carboxylase and fatty acid synthase complex. We have studied 3 steps, that is transport of acetyl coenzyme A, acetyl coenzyme synthase and fatty acid synthase complex, that is FAS complex. Now stage 1, transport of acetyl coenzyme A, source is pyruvate dehydrogenase. How the acetyl coenzyme is transported? For this we require a TCS transporter. Pyruvate is converted to acetyl coenzyme A. How? Pyruvate dehydrogenase and oxaloacetic acid citrate, it is removed from this is the our the mitochondria. This is the mitochondria, from it is being removed. Citrate is formed. Citrate in presence of ATP citrate lyase, it is converted to acetyl coenzyme A and oxaloacetic acid. So our main product which I want to form is acetyl coenzyme A. The transport of acetyl coenzyme has now come to the cytoplasm. First step is done. Now second stage. Second stage is acetyl coenzyme A in presence of A, B and C, that is ATP, biotin, that is vitamin, C is CO2 converted to manolyl coenzyme A. So what I am saying is second step, acetyl coenzyme A is converted to manolyl coenzyme A in presence of ABC, ATP, biotin and CO2. Enzyme used acetyl coenzyme A carboxylase, and this is the rate limiting enzyme. Second step is rate limiting enzyme. Now we'll talk about third step. This is the accept like structure. Accept like structure, that is, this is the reduction unit, upper reduction unit, this is releasing unit, this is condensing unit. We have already discussed this. And this is a multifunctional enzyme because it has many things such as a condensing unit, reduction unit and releasing unit. It is one thing have all the things. So condensing unit, reduction unit and releasing unit. Condensing unit, we have two things, that is manolyl acetyl transcyclase MAT and keto acyl synthase, that is KAS. Second is reduction unit, that is keto acyl reductase, dehydrolase and enolyl reductase in plus acyl carrier protein, that is vitamin B5. Third is releasing unit, that is thioesterase. Steps are like steps are like first of all we have this first step, second this is the third step. This is fatty acid synthase. This is how this was our substance acyl and manolyl formed to acyl manolyl, and in the form of keto acyl synthase it is converted to acetoacetyl residue. It is converted to, in presence of keto acyl synthesis it is formed acetoacetyl residue. Now what happens is the first step of condension, second step is reduction. Hydroxyl acyl residue is formed in presence of keto acyl residue, where the keto acyl reductase, there is the process occur, and that process is NADPH is converted to NADP plus. Now in hydroxyl acyl residue it is converted to it is formed that hydroxyacetyl, I have written wrong thing here. Acetoacetyl residue is converted to hydroxyl acyl residue. Sorry for mistake here. It must be acetoacetyl residue is converted to hydroxyacetyl reductase. Now hydroxyacetyl reductase is in presence of DH, that is dehydrogenase. I admit my mistake here. It was acetoacetyl reductase is converted to, in presence of ketoacetyl reductase, converted to hydroxyacetyl reductase. Now in presence of dehydrogenase, hydroxyacetyl reductase in presence of dehydrogenase is converted to unsaturated acyl residue. In presence of enoyl reductase it is converted to acyl 4 carbon compound. And due to multiple, now the third step is repeated condensation unit. It is converted to 16 carbon compound in presence of coenzyme A synthase, that is this is being removed, and acyl coenzyme, that is 16 carbon compound. What is 16 carbon compound is palmitic acid is being formed. So you have to write this only in exam. What are the steps? Acyl manolyl is there, not attached, then it is attached, then it is in the presence of keto acyl synthase converted to acetoacetyl is converted to hydroxyacyl reduce in presence of dehydrogenase converted to unsaturated acyl residue. Now enoyl residue is converted to by the again and again repeated condensing unit converted to 16 carbon acyl, and now in presence of coenzyme A synthase converted to acyl coenzyme A. Now we will talk about the allosteric regulation. Now we were discussing this is how we have to write. This is available in my Telegram channel, so you can download from there. So now we are talking about what? Yeah. What are the cofactors are required for this fatty acid synthesis? There fatty acid complex, for fatty acid complex we require NADP, as already discussed, and manganese. And manganese are required. NADPH and manganese are required. For acetyl coenzyme carboxylase, that acetyl coenzyme carboxylase is here. Acetyl coenzyme carboxylase, which is biotin is required. So we will see biotin is required, bicarbonate for CO2. How CO2 will come? It will come from bicarbonate, and third is ATP. What are the things required in second step? That is acetyl coenzyme carboxylase, biotin, bicarbonate and ATP. Regulatory enzyme acetyl coenzyme carboxylase. A related enzyme is acetyl coenzyme carboxylase. Second step we have talked about acetyl coenzyme carboxylase. And now we'll talk about allosteric regulation. Allosteric activator, which are the activators? Citrate is activator, and product long chain fatty is 16 carbon compound. If 16 carbon compound is formed, then it will act as allosteric inhibitor. Allosteric inhibitor is a long chain, the product which is formed is the inhibitor. Now we'll talk about hormonal regulation. Hormonal regulation, that is covalent modification, is there well in fed state and fasting state. We'll talk by in two stages, in fail stage and well fed state, and another fasting stage. In well fed stage, insulin is more, insulin by glucagon ratio, the dephosphorylation of acetyl coenzyme, dephosphorylated acetyl coenzyme is there. What is there? Dephosphorylated acetyl coenzyme is there in so if it is dephosphorylated, then activation of acetyl coenzyme carboxylase occur, then fatty acid synthesis occur. In well fed state, you understand, if we are well fed, then there is synthesis of fatty acid because there is dephosphorylated acetyl coenzyme. There is dephosphorylated acetyl coenzyme, so this will activate the activation of acetyl coenzyme carboxylase and lead to the formation of fatty acid synthesis. In what happened in fasting state, insulin by glucagon ratio down, as insulin is less, phosphorylation occur. This phosphorylation of acetyl coenzyme carboxylase. What happened? Phosphorylation of acetyl coenzyme carboxylase. This will lead to inactivation of acetyl coenzyme carboxylase, and this will lead to decrease synthesis of fatty acid. You can understand simply by how in fasting state there is synthesis of fatty acid. Now we'll talk about the triacylglycerol synthase. That is site is obviously liver, adipose tissue, intestinal mucosal cell, and all organ. All which organ we have? Liver, adipose tissue, and intestinal mucosal cell, and all organ. Now organelle, smooth endoplasmic reticulum and mitochondria. Which are the organelle? That is smooth endoplasmic reticulum and mitochondria. Smooth endoplasmic reticulum and mitochondria. What are the site? Liver, adipose tissue, intestinal mucosal cell, and all organ. Now we'll talk about fatty acid synthase. We have already talked. This is how we have to write. So allosteric, this I have discussed. So what are the steps for triacylglycerol synthesis? First step is activation of fatty acid. As earlier we have formed the fatty acid. Activation of fatty acid in presence of acyl coenzyme synthase and thiokinase. In presence of acyl coenzyme synthase or thiokinase, acyl coenzyme synthase, in presence of coenzyme synthase and acyl coenzyme synthase, acyl coenzyme A is formed. Acyl coenzyme is formed by using two high energy phosphate. ATP is converted to AMP, not ADP. So 2 high energy phosphate is used, coenzyme A synthase is used, and the enzyme used is acyl coenzyme synthase, or we can say thiokinase, for the formation of fatty acid to acyl coenzyme. It may be little bit hard for you, but you will see that this simply in fatty acid converted to acyl coenzyme in the presence of acyl coenzyme synthase. An enzyme used coenzyme synthase, and ATP is converted to AMP. No. Second step is activation of glycerol. Glycerol is converted to glycerol 3-phosphate in presence of glycerol kinase. Glycerol is converted to glycerol 3-phosphate in presence of glycerol kinase. Now glycerol kinase absent in muscles, white adipose tissue. Hence in this site, glycerol kinase is absent in muscles. When talk about in muscles, glycerol kinase is absent. And white adipose glycerol kinase is absent. Hence in this side, dihap is converted to glycerol 3-phosphate in presence of glycerol 3-phosphate dehydrogenase. Dihap is dihydroxy phosphate is converted to glycerol 3-phosphate in presence of hydroxyl glycerol 3-phosphate dehydrogenase. By the conversion of NADP is converted to NADP plus. Activation, how glycerol is being activated? Glycerol is being activated by glycerol is converted to glycerol 3-phosphate in presence of glycerol kinase, and glycerol, if glycerol kinase is absent, then dihap is converted to glycerol 3-phosphate dehydrogenase by glycerol 3-phosphate. Product is glycerol 3-phosphate. Now we will talk about the esterification of fatty acid glycerol. Glycerol is converted to glycerol 3-phosphate, glycerol kinase, and dihap is converted to glycerol in presence of glycerol 3-phosphate dehydrogenase, and glycerol 3-phosphate is converted to 1-acyl glycerol 3-phosphate lysophosphatic acid in presence of acyl coenzyme A, it is converted to 1,2-diacylglycerol 3-phosphate, or we can say phosphatic acid. Earlier was lysophosphatic acid, now is phosphatic acid. Now in presence of phosphohydrolase, it is converted to 1,2-diacylglycerol, and in presence of acyl coenzyme A, it is going to triacylglycerol. This esterification fatty acid to glycerol, esterification happen glycerol to triacylglycerol. Esterification happen glycerol in presence of glucokinase converted to glycerol 3-phosphate. Now in presence of acyl coenzyme it converted to lysophosphatic acid. In presence of acyl coenzyme converted to phosphatic acid. In presence of phosphatic acid, we will for writing I have written another. So you can see from there. A storage organ where it is produced most abundant in adipose tissue. This is triacylglycerol is most abundant in adipose tissue. Now we will talk about the cholesterol synthesis. Cholesterol synthesis is not a metabolic fuel. Cholesterol is not a metabolic fuel and exclusive animal sterol. It is present exclusive in animal. It is not present in plant. Okay. Functions of cholesterol: anabolic role means adding up role. Anabolic means adding up. Catabolic means breakdown. Catabolic means breakdown. Anabolic means adding up. So anabolic role is estrol hormone synthesis, vitamin D synthesis. Estrol hormone synthesis, another is vitamin D synthesis through excretion through bile acid. Sites: what are the sites? Organ: liver, adipose tissue, adrenal cortex, and gonads. Gonads, are you understand that? What are the sites? Organs such as liver, adipose tissue, adrenal cortex, and gonads. Organelle are cytoplasm and endoplasm reticulum. Which are the organelle where the cholesterol synthesis occur? Cholesterol synthesis occur in cytoplasm, endoplasm reticulum, and organs are liver, adipose tissue, adrenal cortex, and gonads. Now, substrate. Acetyl coenzyme form private. Now cholesterol is being formed. Starting we have discovered cholesterol is formed. So acetyl coenzyme cholesterol is formed. Cholesterol can be used in the synthesis of vitamin D, steroid, and bile salt. Cholesterol can be used in synthesis of vitamin D, steroid hormones, and bile salt. Note, thiolase is reversible. The thiolase is reversible. Actually, what happens? Statin is basically what happens in body. Cholesterol is being formed. So to statin, how actually statins help? Statin actually competitively inhibit the HMG coenzyme reductase. Statin do? It competitively inhibit the coenzyme A synthase and coenzyme reductase. Coenzyme A, HMG coenzyme reductase. I will. So this is the structure. For this structure, you have to understand there are 3 6-member ring and 1 5-member ring, and this is 8-side carbon chain. This is 8-side carbon chain. 1 2 3 4 5 6 7 8 8-side carbon chain. If you not able to, this can come in your identifying which structure is this. So you can say this is a 8 carbon compound, that is cholesterol. This is 3 6-member ring, 3 6-member ring, and 1 5-member ring. So you can understand the hydroxyl at third position. So this will have to understand which structure is this. So for synthesis of cholesterol, this is the step. This is very longitudinal step, but you may understand this. This is very easy. There are basically 5 steps. What happened in each step we will discuss each by one. This first step is synthesis of HMG coenzyme. Second step is synthesis of malonate. Second step is synthesis of malonate. Third is synthesis of isopentenyl. Fourth is synthesis of squalene, and fifth is synthesis of cholesterol. What are the steps? Synthesis of HMG coenzyme. Second is synthesis of malonate. Third is synthesis of isopentenyl. Fourth is synthesis of squalene. Fifth is synthesis of cholesterol. Acetyl coenzyme A, in presence of acetyl coenzyme A, thiolase is converted to acetyl coenzyme A, and 4 carbon acetoacetyl coenzyme A. Again what happens? Acetyl coenzyme A, 2 carbon compound is converted to 6 carbon compound at HMG coenzyme synthesis. What actually happens here? Here's only I have discussed about statin competitively inhibit. Here's only statin inhibit this. So statin competitively inhibit the HMG coenzyme reductase. HMG coenzyme reductase it inhibit, then there is no formation of cholesterol. What we are doing, the statin is doing, it is stopping the formation of cholesterol. So how it can reduce the formation of cholesterol? By competitively inhibiting the HMG coenzyme synthase. HMG coenzyme reductase, it will inhibit, then there is no formation of cholesterol. So this was the clinical point how statin work. Now We have discussed till now that HMG coenzyme A is formed 6 carbon compound HMG coenzyme reductase in presence of HMG coenzyme reductase. Second step is synthesis of mevalonate. So mevalonate is formed. Now third step is synthesis of isopentenyl. Isopentenyl in presence, there are multiple steps, but in short we can say that 3 ATP is used to form 3 phospho 5 pyrophosphomelanate in mevalonate 6 carbon. And now in CO2 is released and by decarboxylation step isopentenyl pyrophosphate is formed. Another is dimethyl allyl dimethyl. Fourth step is synthesis of squalene. Fourth step is synthesis of squalene. Synthesis of squalene is done by the acids here. First of all, isopentyl pyrophosphate is 5 carbon compound. Isopentyl pyrophosphate is 5 carbon compound in presence of dimethyl allyl pyrophosphate. It is converted to 10 carbon compound. You can see here. It is converted to 10 carbon compound. The geranyl pyrophosphate, dimethyl allyl pyrophosphate, and this isopentyl phosphate 5 carbon compound. It is converted to geranyl pyrophosphate. That is 10 carbon compound in presence of the isopentyl unit, that is 5 carbon compound. 10 plus 5 is equal to 15. Now 15 carbon compound is being formed here. 15 carbon compound is formed here. Then again ferranyl, again with another reaction, there another ferranyl pyrophosphate, that is 15 carbon compound, that will lead to formation of squalene, that is 30 carbon compound. If you didn't remember this, then step must be remembered. First step is synthesis of HMG coenzyme, second is mevalonate, third is isopentyl compound is formed. Fourth step is synthesis of squalene. How is synthesis squalene? 5 carbon compound attached, then formation 10 carbon compound. 10 carbon compound is geranyl pyrophosphate in presence of isopentyl unit. In presence of isopentyl unit is converted to 15 carbon compound, that is ferranyl pyrophosphate. Now ferranyl pyrophosphate in presence of ferranyl pyrophosphate from another reaction, 50-50 and 30 carbon squalene is formed. In presence of epoxide, that is cyclization cyclic epoxide is formed. Then formation of lanestrol, zymostrol, desmostrol. Lanestrol, zymostrol, desmostrol, formation of cholesterol, that is 77 carbon compound. This is the synthesis of cholesterol. This is obviously a longitudinal, but you have to understand that how this is formed. First of all, first step is HMG coenzyme synthesis, second is mevalonate, third is isopentyl, fourth is synthesis of squalene, fifth is synthesis of cholesterol, that is 27 carbon compound. Cholesterol is how much carbon compound? Tell me. 27 carbon compound. So now regulation of cholesterol metabolism. How the regulation we can do the regulation? This is the rate limiting enzyme HMG coenzyme reductase. Feedback regulation. Dietary cholesterol, if the dietary cholesterol is there, repress HMG coenzyme reductase. HMG coenzyme is reduced, then there is no synthesis of cholesterol. This is the feedback regulation. Another is hormonal regulation, that is covalent modification. Hormonal regulation is by in well-fed state, insulin में glucagon ratio is more. Dephosphorylation of HMG coenzyme reductase occur. Activation of if there is dephosphorylation of HMG coenzyme, then activation, and if in phosphorylation it will deactivation. In dephosphorylation HMG coenzyme reductase occur, that will lead to the activation of HMG coenzyme reductase. And now there is increase in cholesterol synthesis. In well-fed state, obviously there will increase in cholesterol synthesis. How? Because there is dephosphorylation of HMG coenzyme reductase, and this will lead to the activation. Dephosphorylated is there, so this will lead to the activation of HMG coenzyme synthase, and this will lead to the increase in cholesterol synthesis. What are the derivatives of cholesterol? Cholesterol derivatives are basically the bile acid excretory form, bile acid obviously excretory form, vitamin D and estrol form. We have already discussed about the three things which are being formed: bile acid, vitamin D and estrol form. Estrol form, that is steroid form, that is mineralocorticoid, glucocorticoid and sexocorticoid. There are three things: mineralocorticoid, glucocorticoid and sexocorticoid. First of bile acid synthase. Bile acid synthase in the primary bile acid and secondary bile acid. Name and site of synthesis. Primary bile acid are cholic acid, and this is a major. Cholic acid is the major. Cholic acid is basically major, and cholic acid. And now we will discuss about the secondary bile acid. Secondary bile acid is deoxycholic acid and lithocholic acid. Deoxycholic acid and lithocholic acid. Site of synthesis is intestine. So I am talking about bile acid synthase. Bile acid synthase is name is cholic acid and deoxycholic acid. Site of synthesis is liver. Secondary bile acid is deoxycholic and lithocholic acid in intestine. Now, what are the stages of bile acid? What are the stages of bile acid synthesis? Bile acid synthesis are first stage. They are divided into four stages. That is synthesis of primary bile acid, secondary conjugation with glycine. First is synthesis of primary bile acid, secondary conjugation with glycine. Third is synthesis of secondary bile acid and enterohepatic circulation and excretion. Therefore, synthesis, conjugation, synthesis, circulation. Synthesis of primary bile acid. Primary bile acid is formed. Second is conjugation with glycine. Third is synthesis of secondary bile acid. Fourth is enterohepatic oxidation and excretion. Now, steps. Cholesterol in presence of 7 alpha hydroxylase rate limiting enzyme is converted to 7 hydroxyl cholesterol. In the mini step, it is converted to cholic acid. Cholic acid is major, deoxycholic acid is minor. So the conjugation in presence of glycine and taurine conjugation, J is conjugated bile acid. This conjugated bile acid and the formation of tyrocholate and glycocolate. The formation of tyrocholate and glycocolate. This is common bile duct. First step is done. That is common bile duct is formed. Cholesterol in presence of 7 alpha hydroxy 7 hydro OH cholesterol is formed. Cholic acid is major and this will lead to the form conjugation of bile acid and tyrocholate and glycocolate, the formation of common bile duct. This is in the liver. Now another step is in the intestine. So another step is they goes to intestine. Now again in liver what happened? Cholic acid, and we talk about the deoxycholic acid. Cholic acid is converted to deoxycholic acid and deoxycholic acid is converted to lithocholic acid. As you know that deoxycholic acid is measured as cholic acid was measured. So deoxycholic acid is measured, and the deconjugation dehydration occur, and then 98%. This is lithocholic acid is minimum. So enterohepatic circulation is there, and then go to the liver. Hence, bile secreted into intestine has 1 degree, 2 degree biles at least amount lithocholic acid. Least amount in lithocholic acid reason is that because it is in minimal amount. Least amount is in lithocholic acid, it is minimum amount. Now we'll talk about bile acid regulation. Rate limiting enzyme obviously is 7 alpha hydroxy. This is first obviously not, but you have to understand that 7 alpha hydroxylase is main enzyme. Actually, what happens if acid pool increases, then the positive impact on FXR, that is farnesoid X receptor, that will lead to the suppression of 7 alpha hydroxylase. Then they will decrease in the bile acid. For example, this is the rate limiting enzyme. If this is being reduced, if this is being suppressed, then there will be synthesis of bile synthesis. Now, what I will say that this will lead to the suppression of bile synthesis. If questions come on cholesterol, you will write this, then you will be get position. So cholesterol is there in presence of 7 alpha hydroxylase, vitamin C and ADP. It is converted to hydroxyl cholesterol. It is converted to hydroxyl cholesterol. Hydroxyl cholesterol, it is converted to two compound. Major is cholic acid, another is deoxycholic acid, another is deoxycholic acid. The conjugation, that is glycine and taurine. In common bile duct, it is converted to cholic acid and deoxycholic acid. It is converted to cholic acid and deoxycholic acid. As this is a measure, it is converted to deoxycholic acid and lithocholic acid. So enterohepatic, it goes this all deoxycholic and lithocholic acid 98 to 95% goes to the enterohepatic circulation. So now this step is regulation. 7 alpha hydroxylase bile acid pool. If bile acid pool is increased, FXR is activated. If FXR is activated, then this lead to the suppression of 7 alpha hydroxylase gene. Then this lead to suppression of 7 alpha hydroxylase gene. Why there is the 7 alpha hydroxylase gene suppression? Because FXR is activated. FXR, what do it do? It suppress the 7 alpha hydroxylase gene. So there is less synthesis of bile acid. So this is how it is being regulated. First step is First step is synthesis. Mm, process how it goes, then it is a regulation. Hope you understand this. I know that I have done in fast way. I don't fast because if I will do in slow way, na, then this maybe too long. I actually is going, but I think that you have understood. Hope so. I'm just quick recapping. It is we talk about the fatty acid, cholesterol, and bile acid synthesis and regulation of bile acid. Overview, fed state, fatty acid synthesis, where it done. I have said two questions. Another thing, I will tell later. The important questions for this topic, I will tell you. Organs is the organs and organelle substrate. This is the step. This is step how acetyl coenzyme comes out, because the reaction is going to happen in cytoplasm, so the acetyl coenzyme has to come out of the mitochondria. So second step acetyl coenzyme carbo. Third step all of us know that the fatty acid synthase complex, that is fatty acid synthase complex is accept like a structure, and reaction by condensation, reduction, and the repetition, then there is formation of palmitic acid, that is 16 carbon compound is being detached from the accept like a structure, that is a fatty acid synthase complex. This happens. I am telling fast, I think so. Please cope up with this. So triacylglycerol synthase, another triacylglycerol synthase. Fatty acid, three stages: transport of acetyl coenzyme synthase, and third is fatty acid synthase. This way you can, if you write enzyme to better. So the why it is called multifunctional enzyme. Multifunctional enzyme, condensing unit has condensing unit, reduction unit, and releasing unit. This has been done, and they for triacylglycerol synthesis, three step again, that is activation of fatty acid, activation of glycerol, activation and esterification of fatty acid to glycerol. Esterification of fatty acid to glycerol is third step. Second step is activation of glycerol, and first step is activation of fatty acid. Cholesterol synthesis, and this is so this is the steps of the synthesis. This is the step of synthesis of cholesterol. Now we talk about regulation of cholesterol metabolism, hormonal regulation, as well as now derivatives and steps of cholesterol formation, of how the in bile, how it happens, and an important question I'll tell you at last. Cholesterol synthesis you can see, and the important question is this only. This can the important question which you can see in your exam, that is cholesterol synthesis, fatty acid synthesis, fatty acid complex as we have discussed, bile acid synthesis, compound derived from cholesterol, and triacylglycerol synthesis. These are the 4 and 2 questions. 6 questions are the important question. Cholesterol synthesis, fatty acid synthesis, FS complex, bile acid synthesis, that is how bile acid is formed and there is its regulation. Compound derived from cholesterol, 3 compound, estrol, and we have discussed now. These are the steps of cholesterol synthesis, fatty acid, and this can become an enzyme. And this hopes you enjoyed this video and like this channel and subscribe. Like this video and subscribe to the channel. Bye.